Understanding about the start time. So I'm going to talk to you a little bit about biogeochemistry of coccolithophores and their optical properties. And let's see if I guess this pointer, does that pointer not work? Is there another? No. Am I? Oh, okay. I got it. Yeah. It's all right. So. Oh, that's even better. Excellent. Okay. So, little bit of an outline. I'll talk briefly about the taxonomy, aspects of blooms and their physiology. Who cares? Reasons you should care about these from biogeochemistry, ballasting, impact on the carbon cycle. It's more of the big picture stuff. And then different aspects of their optical properties and ways to measure them. So coccolithophores are a group within the marine planktonic calcifiers. And they're probably over half of the marine planktonic calcifiers. But there's some other important ones here, not from an optical standpoint, but just from a mass flux standpoint. The foraminifera, and so these are 50 to 500 microns, and the pteropods, which are well-known salmon food. We'll talk a little bit about ocean acidification, and these guys have major issues with ocean acidification. But generally, it's felt that coccolithophores represent over half of the marine calcifiers. They're in this class called Primnesiophyceae, and within the class, there's a family called Haptophyta. These are scale-forming algae, and the family Haptophyta produce scales that are made of calcium carbonate. They're evolutionarily pretty young, less than 2.5 million years. All the species that we have on Earth now made it through a couple of bottlenecks in evolution, the Cretaceous tertiary boundary being one, and there was another bottleneck in the Ordovician, but basically, and E. huxleyi in particular is very young. They come in all shapes and sizes. This guy here, Barutosphaera bigelowii, there's been some really cool recent things that have been happening with this. This thing has different life stages associated with its sexual cycle, and apparently it's cued into the lunar cycle when this thing decides to have sex. And these others, you can see these various shapes. Here's Coccolithus pelagicus, which has two different types of, it has two life forms. One which is called a holococcolith and the other which is a heterococcolith. And these guys were originally thought to be different species. Because they were looking at them in the geological record, and they said, well, the species that produces this coccolith can't be related to this one. But in terms of their life cycle, one is haploid and the other is diploid in terms of the amount of DNA that they have. And here's our old friend Emiliania huxleyi, which is kind of the star of the show for a lot of the spectacular optical events that we see from space. And here's another shot of this guy, Coccolithus pelagicus. And some more shapes and sizes. Some of them, not all of them, but some of them drop their coccoliths constantly. And I call it like an oceanic dandruff that is found everywhere in the ocean. It's virtually impossible. Every place I've looked, I've found them except south of the polar front in the Southern Ocean, and up in the Arctic, they're pretty difficult to find. Some more observations about these. They're a major part of the sediments of the Earth. It's about 1/4 of all sediments on Earth come from coccoliths. And the first observations, we'll talk about these in a second, go back to the 30s for the blooms. So here's one of my favorites, this one they called Coccolithus huxleyi summer. The old scientific name Emiliani huxleyi was Coccolithus huxleyi, and the name was changed actually after a gentleman named Cesare Emiliani, who discovered some fascinating geological cycles that were based on the preservation that he found based on microfossils of coccolithophores. Berhe came up with this paper in which he was looking at discoloration. So this goes back, this is the 30s, I believe, and they were seeing these massive decolorations. June of 1955, they were seeing, oops, let me go back. They were seeing concentrations up to 115 million cells per liter. And another paper which I've really come to grow and appreciate is about the distribution of these things, is a real old chestnut. It's a classic paper from 1967 by McIntyre and Bay. And these guys went out and they sampled the Atlantic principally in the water column and the sediments. And then they compared the coccolithophore species they found in both. And the idea was, does the distribution in the surface waters reflect what we see in the sediments? And in fact, it did not. There was a narrower distribution in the sediments than there was in the water. Some of these things were expanding their range. This is a recent picture of Andy McIntyre. He's still with us and going strong. So what they defined were 5 different zones in the Atlantic, and you can see all their stations here. They had a tropical zone, 2 subtropical zones, transitional zones, which refer to the subtropical front, the position of this. This is a major frontal feature, and then a subarctic zone, which is number 4, and a subantarctic zone here, which is number 5. And I note for you that south of the polar front, where I said I've never found any coccoliths, they didn't either. So their floral zones here pretty much end at the polar front. However, in the Arctic, they didn't have any stations higher than that. They still had stations where they found coccolithophores. And we've since been up to about 75 north in the western Arctic, and we've found coccoliths under the ice and at the ice edge and 100 kilometers into the ice. So they're even up there. Now, one interesting aspect, I don't expect you to read all these names, but the diversity of these decreases towards the poles. So as you go from the tropical regions, you see all these species here, and then as you get up to the subarctic and the subantarctic, there are 2 species left. And that's not quite right, at least from what we've found in the Indian sector. There are certainly more than 2 species. But the other thing that's kind of cool about this is this one species, Coccolithus huxleyi, back in 1970, really Emiliani huxleyi, are found in all of these floral zones. So they're basically ubiquitous. And that's one of the amazing things about the species. It has found a niche in just about every possible marine environment. So what is a coccolithophore bloom? Well, these blooms in 1983, which is the first reference, this is a paper in Nature, and it's the first reference to a space, a bloom observed from space. If you go Googling it, you'll find that all references point to this particular citation. And they saw 8,500 cells per mil, or 85,000 per liter, and 78,000 coccoliths per mil, or 78, forget it. I can do the milliliter calculations here. These are really dense. And note the chlorophyll in these can be quite low. That's one of the weird things. It's a misnomer about coccolithophore blooms, People talk about different algal blooms, and they say, oh, there's so much biomass of coccolithophores in there, but there's not. In fact, typically these blooms will have 1 milligram of chlorophyll per meter cubed in there. And so in terms of their impact on the biogeochemistry, it's more related to the amount of calcium carbonate that they produce. But it can represent a significant discoloration. So here's that paper. Holligan, Violier, Harbour, Camus, and Champagne-Philippe. And they were using the coastal zone color scanner. And that was the image from 1979. And these bright features here on the edge of the Western European continental shelf were what they were going out to figure out because they could see them. And when they got there, this is what they found. This is actually a fecal pellet of a copepod, but it's full of coccoliths, and you see lots of detached coccoliths over here. And that's a scale bar. It's 10 microns probably, or yeah, 10. So this was a big deal when they discovered these. Now another aspect about coccoliths, before we talk about their optical properties, if you take a whole bunch of sediment trap measurements from around the world, this is from Roger Francois, and you look at the flux of calcium carbonate to the flux, to the fraction or the transfer efficiency of POC, so organic carbon, going to depth. So this is the organic flux divided by the export flux, and this is the total flux of calcium carbonate. They're correlated. And now sediment traps are notorious for having complications, and there are all sorts of issues. And so if you can get a regression like this with the 80-some sediment traps from around the world showing that there's this relationship, that's kind of interesting. And what they argued was that this flux of calcium carbonate represented ballast for all of this stuff, and that because it's dense, that it was in effect ballasting the sinking particular organic carbon to the depths. That is not uniformly accepted. There's some other explanations about what moves sediments to the deep sea. I don't have time to go into them here, but this is the ballasting hypothesis has gotten a fair amount of attention over the years and is still talked about. So this is, in effect, the efficiency of the biological pump. So half of the biological pump is removing half of the anthropogenic CO2 that we have put into the atmosphere. This is why this is important. So if we didn't have this biological pump, the atmospheric CO2 would be a lot higher than it is right now. Okay, a little bit about calcium carbonate and global carbon pools. As I said, it's calcium carbonate is responsible for about 1/4 of all marine sediments. There are many calcifiers, but the small ones play a disproportionately large role in the carbon cycle. And I put the pool sizes here for PIC, DOC, POC, so particulate inorganic carbon, that's calcium carbonate, dissolved organic carbon, particulate organic carbon, and atmospheric carbon, and these are in gigatons. And so you can just get a feeling for the relative size of these different pools. And you see with 1/4 of all sediments in the ocean coming from calcium carbonate, that's a pretty darn big pool. Now I have to tell you a little bit about some stoichiometry here of calcification, because that's another, to answer the question of who cares, I have to describe this. So essentially what's thought about the process of making these calcium carbonate shells, the organisms are fundamentally taking 2 bicarbonate molecules here, combining them with calcium and seawater. They're generating 1 molecule of CO2, some of which can go into the atmosphere, some can go into photosynthesis, and 1 molecule of calcium carbonate in water. And in the top kilometer of the ocean, so it's generally considered to be saturated for calcium carbonate, this reaction is strongly driven to the right. And, but there are factors like pressure, temperature, that can affect this equilibrium. So the deeper you go, for example, the higher the pressure, the more soluble calcium carbonate becomes, and it actually goes back to bicarbonate. Marine calcification is thought to be about 1 to 1.5 gigatons per year. So that's a fifth of all the fossil fuel CO2 generation. Actually, less than this, less than that now, because we humans keep producing more and more fossil fuel. We keep burning more and more fossil fuel, and but it still is equivalent to about the CO2 production associated with deforestation and tilling of soils. And so what that means is that if you've got 1 to 1.5 gigatons of calcification calcification happening each year, and you've got 1.5 gigatons of CO2 going into the atmosphere, you have to put that into your balance, into your budget. So I'm going to show you a little bit from the Gulf of Maine, a little closer to home. In the late 1980s, we were getting these high reflectance features in the Gulf of Maine. And we had no idea what they were. We had all seen Holligan's paper. And we thought maybe these are coccolithophorids. And so in the late 1980s, we didn't have the coastal zone color scanner. It had died. And we didn't have any ocean color instrument in space. So we had to resort to using this advanced very high resolution radiometer, AVHRR, which is used for measuring temperature. But it has one broadband visible channel, which is not the most sensitive channel in the world, but we were able to see bright coccolithophore patches. And so in the Gulf of Maine, we were seeing things like this. And so we called up the funding agencies and said, hey, there's this bright patch of something out in the Gulf of Maine. Could we get a couple of days of sea time to go out and look at it? And fortunately, they gave us this time, and out we went. And what we saw was basically water, this turquoise color. Now, this is taken from a photo. I was wearing a blue, navy blue jacket there, so for comparison, you can see the color. It was like you had run aground in Bimini. And another picture, and that's Steve Ackelson, Patrick Holligan right there, and that's me. And that's the navy blue color there. And again, you can see the color of this feature, and you can even see a boundary here off in the distance. It was a spectacular event. And when we filtered the water, that's what we found. You're hard-pressed to find a particle that's not a coccolith in this. Right there, that's one maybe. So just when we thought these blooms couldn't get any bigger, we were doing a trip in the North Atlantic, and we went through this feature right here. And these are a series of, this is No 11, AVHRR. These are a series of images made over about a week or 2-week period. And we were steaming basically through this eddy right here. And we were doing a section through it, and then we were tooling around, trying to understand something about the calcification rates and the light scattering. And so the area of this thing was 500,000 square kilometers. It was truly one of the most magnificent events of my career, being out in this thing. And you can see, if you look carefully, there are some non-calcite eddies, and there's a calcite eddy here, and just how complex the waters are in this part of the world. You know, you'd be hard-pressed to describe that with just saying there's one water mass here and one water mass up there. So coccoliths were in some ways allowing us to visualize the complex hydrography. Here's an old picture of chuck trees. This is outside the bloom, inside the bloom, and you can see that I'm using his color chip sweatshirt here as a comparison. And while others were looking down, some of us were looking up, and this was a flight coming over from Glasgow, Scotland, as it turned out. And we thought, wouldn't it be cool if we could call up this pilot? They're usually taking a nap during this time of the flight, and maybe see what it looked like outside the window. And so we raised this guy on the crew of the Darwin, the ship, the Darwin, raised the pilots on an emergency band and asked them if they could look out the window. And this guy, it was a Lufthansa flight, and with a very Germanic accent said, "Oh, just a moment." You could hear him putting his seat up, and he looked out the window, and we could hear him exclaim, "Oh, this is incredible. I've never seen anything like this." And this is what he saw. And this is one of those eddies. We were that white dot, I think, right there. And this is one of those giant eddies. And we asked if he had a camera and he could take a picture. And he said no, but he would radio back into the cabin. And so he radioed back into the cabin, found someone with a camera, and brought them up to the cockpit. This is all pre-9/11. And they took this picture, and then 2 weeks later, this envelope showed up in the mail, and this is what was in the envelope. It was very cool. But it just gives you a different appreciation for the scale of this particular, of the feature, really. Okay, so Toby Tyrrell did a wonderful paper on sort of the overall, the big picture of what coccolithophorids do to the water column. And he had this figure 2 in which he said, okay, take 100% of the incident radiance, and he had 2 situations, no PIC or calcium carbonate or 300 milligrams per meter cubed. And he said, okay, let's just look at the relative proportions of where light goes coming in, same situation, modeling this. So in a situation with no calcite, 0.4% is scattered out, 6.5% overall is reflected, 81% makes it down one attenuation length, 7% or 8% is absorbed by chlorophyll, and 4% gets transmitted to greater than 20 meters. Now if you add this amount of calcite, and this is a typical amount for a high density bloom, you would have 5.5% scattered, 6.5% reflected, and 80%, not much difference, absorbed by water and other things, 7.6% absorbed by chlorophyll, but 0.5% as opposed to 4% transmitted greater than 20 meters. So it's basically scattering all this light in the top layer, lots of multiple scattering here, and much to the detriment of things that are living below there. And so this suggested to some people, well, perhaps what coccolithophores are doing is they're just providing a canopy and they're shading out everybody down deep and keeping all the light up here in the surface layer to themselves. So that's one, when people say, well, why did these things make coccoliths? Perhaps one is related to competition for light. In the Gulf of Maine, blooms usually begin in moderately stratified water near the summer solstice. And this got us thinking, well, maybe there's some sort of factor in the release, some biological factor related to the release of the coccoliths near the solstice, because quite frequently it was just the week before the solstice when we would start seeing these. Here's an example. This is an early MODIS image of a high reflectance feature that was off the southern flank of Nova Scotia and was wrapping around Jordan Basin. This is called Jordan Basin out here. And it was forming right at the edge between well-mixed water and stratified water here, and which is typically the spot where you expect to see coccolithophorids. Diatoms are thought to prefer mixed environments, and coccolithophores partially mixed environments or moderately stratified. And if you look for the distribution of coccolith concentration plotted here against density, so this is 21 to 25 sigma theta units, and day length, so the solstice is the longest days of the year, you see these blooms are occurring just at about the longest day length. And that's not always true. We can see blooms earlier than that further south, so I don't honestly think that it's a biological component to them dropping the liths. I think it's more related to the seasonal mixing stratification. So, some optical properties we're going to talk about. Optical properties of PIC itself has a relative refractive index of 1.19, and particular organic carbon, so what the rest of the stuff in the ocean, biogenic material is made of, has a relative refractive index of 1.05. So big difference there. These are big scatterers. And biogenic silica, which is what diatoms make their frustules out of, only has a relative refractive index of 1.07. So while we have seen examples of high reflectance features from space that are indeed diatom blooms. So one example is in the Bering Sea. We saw a winter storm in February that had mixed up all sorts of diatom frustules from the sea floor, and it kind of looked like a coccolithophore bloom. But with that kind of relative refractive index, it takes a lot more silica, biogenic silica, to make the same signal as PIC. Dense ocean suspensions of coccoliths can have a high albedo, 35%. That's a lot. PIC is birefringent. It rotates the plane of linearly polarized light by 90 degrees. We use that when we enumerate them. It has low absorbance. And the mass and shape of these coccoliths, I've shown you some examples. It varies by species, and hence the scattering cross-section, which is in meters square per mole, is variable with values ranging from 1 to 8 meters square per mole. And that, by the way, is very important in terms of how we design an algorithm to sense them from space. And coccoliths can be a primary determinant of normalized water-leaving radiance. So here is what I base that comment on. Here's normalized water-leaving radiance measured above the water from our ships of opportunity. And here is the concentration of PIC. These are all from the Gulf of Maine. And these are different wavelengths: 412, 443, 490, 510, 555, and 683. And the thing to note about this, here at 510, just looking at normalized water-leaving radiance, we're not looking, and comparing that to calcite, we're accounting for 42% of the variance, which, you know, if you were to regress chlorophyll versus 440 nanometers, you might not see as high an R square, just at a single wavelength, because there are other things that are absorbing light in that waveband. And here, you know, this is, here we see 37%, 31%, and even in the red, it's not a great relationship. But these things, this oceanic dandruff is reflecting light and is a primary determinant. Yes?
Just go back to that.
Yes.
We looked a lot at radiances and these relationships, and they're always on a log plot, and that's what
Yeah.
we apply.
Yeah.
Really appreciating that range of variability.
Thank you. Yes. Well, now you just have said that. Now I'm going to switch to a log plot. Because coccoliths vary over such a large range. Here I'm showing you now, but these are actually, those regressions were made in linear space. But you can see with coccoliths, you know, we actually get a better relationship than we do with PIC. And this is one hint that through, you know, PIC comes in many shapes and sizes. It's not all the same. Coccoliths vary in their size, and there are other calcifying, small calcifying calcite particles. Therefore, there's something about these coccoliths which is unique, which when we measure PIC, we're measuring PIC of everything. There are even some cyanobacteria that calcify, that have calcite associated with them. And there's a paper published not long ago, and we're still digesting this, but there are things that are made of calcium carbonate that are not coccoliths, that may have different scattering cross sections that would gummy up the relationship when you do it PIC versus normalized water leaving radiance. And coccoliths are quite unique in their ability to scatter light, and I'll talk about this more in a second. Okay, how about absorption? Now we played this game where we were looking at the absorption of E. huxleyi. This is from a natural station, and we were using the filter pad technique here, but we were basically one sample we dissolved all the coccoliths, and the other we maintained the coccoliths intact, and basically the only differences we could see were out here in the UV, and basically everything else in the visible was identical. So the absorption related to coccoliths is really related to calcium carbonate is really not very high. Now remember what I said, that all coccolith or all calcite particles are not equal. If you assume calcite spheres and use the Van der Hulst technique or computation, you take the relative refractive index of PIC, its density, micron size coccoliths have the highest. This is a scattering cross section, not a backscattering cross section, but I put down below, so this is diameter versus scattering cross section. Here's your E. huxleyi. Here are aragonite needles, which the sizes of them. Foraminifera tests, remember I talked about foraminifera, and pteropods, which are giants in the calcifying plankton world. Basically, we're orders of magnitude higher scattering cross sections for coccoliths than for foraminifera or for pteropods. And when I saw this, I thought, wow, either evolution has, if coccolithophores are using the optical properties of their coccoliths, it's being selected evolutionarily, they couldn't have found a better size calcifying particle to scatter light. And certainly these guys are not using the scattering properties of calcite related to their survival or their selection. But coccoliths have homed in on this size remarkably well. So maybe there's an optical purpose to these coccoliths after all. The volume scattering function flattens in the backward direction. And this is a paper that Ken published using his general angle scattering meter, and we made barrels of E. huxleyi culture, and then he submerged this thing, and we're looking at the shape of the volume scattering function. And basically, you know, we're looking at this. If you've been looking at volume scattering functions like the Petzold functions, they always go up in the backwards direction. This thing is relatively flat. And we had different types of growth phases represented here just to try to...
What sort of backscattering fraction do you get?
So BB tilde?
Yeah.
3%? Something like that. 4. I mean, 4 is as high as you, right? You don't get above 4, I think. In a bloom, we've seen 2 to 3.
I've seen 4.5% from resuspended quartz sediments.
Whoa.
But that's extreme.
Yeah, yeah. Yep.
Is that they actually they're pigmented, so they're absorbing. So if they were pigmented, they might have a higher scattering.
Yeah, I'm talking about, so the numbers I just gave you were for 530 nanometers, so we're beyond the, but you're right, yeah. The wavelength dependence of backscattering, which is also in, excuse me, in Ken's paper. Typically it's -1, -1.4, 1.2 for either naked cells, plated cells, or detached cocculus. So when you're modeling these, that wavelength dependence for the scattering is a good number. And we played games with flow cytometry. You've probably heard about absorption cross-sections for phytoplankton and how the performance of chlorophyll algorithms varies, declines when you get to high latitudes because of the package effect. You've got all these big cells that have absorbing chlorophyll as opposed to comparing to very small cells in the subtropical gyres. Well, just like there's a package effect for absorption, there is kind of a package effect for scattering. And so what we were looking to do was to use a flow cytometer to sort different species of coccolithophores. We grew them in culture, and then we separated them off into small vials in which we could get high enough concentration to measure their backscattering, and to look at how much that backscattering cross-section varied. And so here were cultures of different coccolithophore species, and you can see the variance here from 2 to 8 with an average of about 6 meters square per mole. And then we took field samples and we looked at the variance there where we were sorting based on their birefringence properties. And we were getting even wider ranges of the scattering cross-section. So, of course, in a natural assemblage, you could have sedimentary particles that were suspended. You could any number of calcium carbonate things, the aragonite needles and such that you might find down in the dry tortugas. So, but that number is highly variable, and that's probably one of the biggest sticklers that we have in the the PIC algorithm. Yeah. Yeah, I'll get to that. Yeah, yep. Next slide. Okay. So not all E. huxleyi cocculus are alike. The folks doing measurements around the world of E. huxleyi have affectionately come to know the type A, type B, type C, and type R cocculus. And they vary in their distal diameter, the size of this internal piece here, and I have a table in a second. Hopefully I didn't take that out. But the variance here in the calcite per coccolith it varies. So let's see if I have, and this is a paper by Alex Poulton. This fifth morphotype BC, which is this guy here, is found in the Southern Ocean, for example, and in subpolar waters, is not as robust as the type A. So, oh yes, good, I kept it. So here, here's the picomoles. I'm sorry, so the type B actually has more calcite than the type A, which is the North Atlantic. And this is 50 to 90% bigger than type A. And this type BC, which is kind of another morph here, which is 73% smaller than type A. So some of this variance, not only is the shape is pretty much the same for E. huxleyi, but the amount of calcite varies. So I think that could have some, yes?
If you did the normalization instead of to the lip area, would that look like?
Yeah, well, so ask Howard Gordon. So he with this paper with Dew was modeling taking Jeremy Young's, who was the guy that described these morphotypes, and he was trying to model the shape, the scattering, based on a geometric representation of the actual shape. So he started with fishing reels, and then he came up with, he had discs and saucers, fishing reels, and those aspects do make a difference. So in those radial elements, these radial elements here, which are 25 nanometers apart, can vary. And you can see here, this is not a terribly well preserved one, but even looking at the back side, you know, there are these are not perfectly shaped. There's variability, and there's species-related variability. But yeah, you cross-section, you'd have to take into consideration. Yeah.
To the size of the lip. So
right.
The PIC, that you could normalize that.
Yeah.
But certainly the shape.
Now, and the other part, which gets right back to the crux of the problem with the PIC, with making a PIC algorithm. Think back to that McIntyre and Bay paper. The diversity of these species increases as you go towards the tropics, and so. And E. huxleyi is uniform through all of these. E. huxleyi makes the big blooms that everyone says, wow, check out the size of this. But in fact, we're trying to do it, we're trying to measure calcite in the ocean at a background level. And in the tropics, where we have 10 different species with 10 different, vastly different shapes than these, the problem gets more difficult. So let's look at the backscattering cross-section. All I'm gonna say now about this term here on this axis is it's the we call this BB prime or acid labile backscattering. I'll tell you how we measure this in a moment. But this is the scattering that disappears when you dissolve the calculus. And so for a number, I'm going to show you now. These are Atlantic meridional transit cruises that go through the central gyres. If you plot the PIC concentration versus this BB prime, you get a slope of 2.2 on average. But these are means, and there's variance associated with those means.
Sorry, question?
Yes.
Those morphotypes, are they genetically distinct, or are they?
Yeah. Yep.
So
Yeah, they are the same species, but they're they are varieties, if you will. Yeah. Here is in the North Atlantic, plotting suspended calcite versus BB star. This is at 546 nanometers, and this last one was at 532. The slope of this guy is 1.6, but you can see the variance. And this is a quasi-unialgal. I mean, there were other calcifying species in there, but and here is from the Southern Ocean, and again we're down in the region of where it's mostly E. huxleyi, and the slope of this guy is 1.5. So at the poles we're doing pretty well, but in the middle area there is going to be more variance. So a couple of aspects about optical ways to measure PIC. Birefringence, acid labile backscattering, and these satellite PIC algorithms. Birefringence, the technique that was used, has been used for years by geologists, and we've adapted it a little bit more recently. It was described Haydar and Tierstein and Deuser. It was used before these guys, but they had a nice paper where they were looking at these in Bermuda at the BATS station, the Bermuda Atlantic Time Series. And this technique involves filtering your sample onto an HA filter, a 0.4 micron HA filter, polycellulose HA. And then you carefully put a drop of this Canada balsam, which is nothing but pine sap, but the chemical companies have figured out, they can harvest this stuff, purify it, and sell it for $100 a bottle. And it's optically very pure. And you put a drop of this Canada balsam on a glass slide. You lay that filter on top, and the filter clarifies. You can actually, you see right through the filter, and you can put polarized light through that then to look at the birefringence. And it's a cool technique also in that you put, after you put the filter on the Canada balsam, you put a cover slip on top, you heat it up to 60 degrees C so it melts the pine sap. And then you've got a slide which will last forever. You know, you put it in a box and you can go back anytime in the future and go back and look at it. There's another technique which uses in situ birefringence. It was published by Guay and Jim Bishop in 2002 as a birefringence method for measuring calcium carbonate. So let's see what. So this is that paper. And basically the idea was they wanted something they could put on autonomous vehicles to measure the birefringence of PIC. And so they said, okay, they took some pure silicate here. This actually involves sodium chloride, calcium. This picture has sodium chloride, calcium carbonate, and silica dioxide. So this is basically sand. And this is under the two polarizers. So now I have to use my hands. So you have a light source underneath that's going through a polarizing filter. Your sample is here. It's the light then polarized goes through that sample, and you have a cross-polarized filter up above it, which is only collecting the light that has been rotated 90 degrees. Does everyone maybe I can use your board for a second. So you have a light source here, and you have a polarizing filter, and I'm just going to describe this with a linearly polarized filter. You've got your coccolith here on your microscope stage, and then you have another polarizer here which is oriented orthogonally, and then you have your objective here and your eyeball looking at the light that's coming through. So your light source comes through, it's letting through polarized light in this orientation. It hits the coccolith rotates at 90 degrees. That light can get through the upper filter and goes back to your eyeball or the sensor. And in this case, they cross the polarizer, and you see all this calcium carbonate. You don't see any sodium chloride. You don't see any silica dioxide. Yes?
This technique is the basis of how they do grain analysis in geology transections. You put transections of rock with calcium chloride
Yeah.
and minerals from the same properties, and also sea ice. You can take sea ice and cut thin sections of ice in the freezer, of course, and put it under cross polarization and identify the orientation of ice crystal growth.
And the geological applications of this way predate the biological applications of this. So Jim's idea was, okay, let's take a beam transmissometer and we'll put a polarizer at the bottom. We'll let stuff settle on it, and if there's any calcium carbonate in there, and we'll have a polarizer, a cross polarizer at the top, and then we can measure this birefringent light. It's a great idea. And I'm going to show you now some examples of birefringence. So these are what coccoliths look like under a microscope, and this is from an Atlantic meridional transect at 48 degrees north. That's a plated cell. Of what species, I can't tell you. This looks most likely like an E. huxleyi coccolith. They have this sort of iron cross behavior. This is not an E. huxleyi because it has a sigmoidal shape. There's a species called Japhira capsa that has a little bridge in the middle of its coccolith, and that kind of looks like that. And there are different species there. Here we go a little further south. So you're looking at a dark field. That's a plated cell. And here's 11 degrees south. Lots of different patterns in birefringence. And here's 33 South in the middle of the South Atlantic Gyre. There's a nice example of a Gephyrocaps coccolith. I don't know if you can see that little bridge in the middle. And 40 degrees south. The only problem with it is that coccolithophores are not the only birefringent particles in the sea. And Jim Bishop and I have argued ad nauseum about this. And here's, to the extent that I published this picture just for Jim in this paper, this is a serratium, a dinoflagellate that has clear birefringence properties. And zooplankton carapaces, lipid droplets, detritus, and even some dinoflagellates can rotate the plane of polarized light. So the way to get around that, it's not a fatal error for the Carbon Explorer. The way to get around that is to simply take your seawater samples, which will include this sort of material in it, and calibrate that instrument to naturally occurring material. So it means your error bars will go up because you're going to be getting birefringent light from birefringent dinoflagellates, but at least you're including these in the calibration. And then you do a totally independent measurement of PIC, like using atomic absorption or flame photometry, or to get a solid number on the PIC such that you can calibrate such things out. So here's how we actually combine birefringence and image analysis to enumerate coccoliths. Believe me, paying somebody to count these things with a clicker for years at a time is expensive, and we had to come up with a better way. And the reason I wear these glasses is because I used to do just that. And these days, there are lots of image analysis ways that you can get at this. And we were funded by NSF to work with a group at UMass who do facial recognition in subways, for example. This is low-hanging fruit for them to try to enumerate coccoliths, not necessarily the different species, but just coccoliths and plated cells using birefringence. So here's an example of an image from a bloom. It's a little fuzzy there, but you can see we're going to zoom in on this particle right here. And now the image analysis so we came up with a program called CCC, which is counting coccoliths. I can't remember the third C, but and basically we look for hills and valleys in the image itself, which represent birefringent light, and then we look at the distances and associated shapes of these to say and the amount of light to say whether or not it's a plated cell or a detached coccolith. So in this case, it's going to be calling these a plated cell, and we'll home in on an example. That's what it's calling a plated cell. That's an individual coccolith on the side. There's another individual coccolith, and these are plated around. Looks like it has multiple layers around that cell. And then the model basically describes, it looks at the shape, looks at the dimension, because we know how big these coccolithophorids are, and the amount of light, and it will make the decision based on those aspects. And now we're going to zoom in on that detached coccolith off on the corner. And this is an extreme blow up, but it's defining the center of those four, that iron cross, and then it measures the distance between them, and it says, well, they're within a certain size range and angle here that we can accept as being a coccolith or not. And so we use this now, and it allows us to basically load up the microscope every night before we go home, and it counts these things all night, and we come back in the morning and load it up again. Okay, acid labile backscattering. This is what I described for you before. This is an underway method that we use as the ship is steaming along for looking at for evidence of calcium carbonate. And so we start with a plot here with axes of time, and there's particle backscattering that's going to appear up here, and pH is going to appear down here when we change the pH to dissolve the cauculus. And every now and then, like every couple of minutes, an acid pump comes on to inject a weak acid into the seawater stream, and we're going to then be dropping the pH and then increasing it, dropping and increasing. So this is ambient pH of seawater, and this is below the dissociation point for calcium carbonate. It's really important that we know we're dropping the pH enough that we dissolve the cauculus, but not dropping them so low that we are dissolving everything in the sample. So we're getting it below the dissociation point for calcite, which is one of the forms of calcium carbonate. And then we turn off the pump and the pH comes back up, turn on the pump, so forth. Now if you follow the light scattering signal, if you're going through a bloom, you would see something like this. When the pH is high, the backscattering is high, it drops, goes up, drops, goes up, and drops with these cycles. And this is when we're in the field and we see this, we're jumping up and down because it means we're going through something good. Much of the time, it's flat. There's no change or very little change. So then we take the average of the peaks and the troughs and we connect them, and then we look at the difference between the trough and the interpolated quantity here at the peak. And that's what we call acid labile backscattering. And it turns out to be a fairly robust way of measuring PIC. There are some problems with it. The ship is steaming along, and so we're not necessarily in the same spot here as we are when we're here. Or we're not in the same spot here. So we're hoping that the ocean isn't changing so fast that we can't say something about the spatial variability.
What's the time scale that you use?
Minutes. So on the AMT, we sample longer periods of time here because we need more signal to noise. So this could be 9 minutes, and so it's 18 minutes, at which point, but you're going through the gyres. But we can get the standard errors way down.
But you couldn't do 2 total all the time.
Then we'd have to intercalibrate. This is using the same backscattering sensor.
But if you had them together, all this, for 10 minutes, and doing 2 different things for 10 minutes.
Then we could, yeah. But yeah, our goal was to avoid having 2 backscattering sensors that we had to intercalibrate. Because the differences could be on the order of 1 e to the minus 6 per meter, which is pretty low in. Yeah. So if you look at ship acid labile backscattering, this is the optical technique, versus where we've actually taken the sample using inductively coupled plasma atomic absorption. Basically, we get a linear relationship down to a certain amount of backscattering, and then below that, we simply can't separate the backscattering, the variances in space, errors caused by variability in space, and so forth. So, here's that 1 e to the minus 6 number. Here, we're getting down to 3 e to the minus 4, say, for the difference between these two. And the lowest value, 5 e to the minus 5, where we can actually make sense of what's going on. And the optical technique that's off the screen is linear down to about 5 e to the minus 5. Okay, remote sensing. There are 2 PIC algorithms. There's one that's based on NLW 440 and 550. We call this the 2-band approach. And there's the 3-band approach, which is based more on red and near infrared by Howard Gordon. And the 2-band algorithm is essentially a lookup table. This is NLW 440 on this axis here, NLW 550. And these isoplests, which is the lookup table, if you will, these are lines, iso lines, for chlorophyll concentration. And the more horizontal lines are isolines of coccolith concentration or converting them to Emiliania huxleyi type coccoliths, that amount of PIC. So this being the PIC, these lines being the chlorophyll. You can see as we go close at lower and lower radiances, which with more and more chlorophyll in the water, we're getting down here. These lines get awfully close together, and our ability to separate chlorophyll concentrations, for example, is reduced. It's enhanced when we're in more oligotrophic waters. And the radiometric error of MODIS is 5%, but